today I will be talking to you about parasites. Over time, unfortunately, parasites have obtained a largely negative reputation, but today I'm hoping to flip that script and convince all of you that parasites are very valuable research tools in science. If you look at the diagram in the top right-hand portion of your screen, you'll see two different phylogenetic trees. The one on the left is showing different species of pocket gopher, and the one on the right is showing different species of louse. The dotted lines connecting these two phylogenetic trees are showing a nearly 1-to-1 ratio between host species, the pocket gopher, and the louse parasites. This is the key for researchers to be able to only sample parasites and still make inferences about the genetic or population structure of hosts. And that's exactly what I'm hoping to do in my study. So if you look in the bottom left-hand portion of your screen, you'll see a gross little tadpole-looking thing, and that is my parasite that I'm studying. It's called Pleurogonius malaclenes, and it is a species of Digenean trematode. Its life cycle is very simply shown by the yellow arrows on the screen. First, it's able to infect a species of mud snail called Tridia obsoleta, and the snail is then eaten by a species of turtle called the diamondback terrapin. Eventually, the parasite is able to reproduce within the terrapin. The turtle will release the parasite's eggs in its poop, and then that is eaten by the mud snails, and this whole process kind of starts over again. So the diamondback terrapin is necessary for the parasite to complete its life cycle, and as a result, they're only found in the same area on the east coast of the United States, from roughly Rhode Island down through Florida. That is a range that currently exists in literature, but the first goal of my study will be to sample snails on Cape Cod in order to establish whether or not Pleurogonius malaclenes is present that far north. The two species it depends on are found there. So my hypothesis is that this species will be found there too, and that I will be able to update the existing literature on the parasite's range. Secondly, I'm hoping to take genetic samples from Tridia obsoleta, the mud snail, and this parasite in order to create phylogenetic trees very similar to this figure on my screen. After doing so, I'll be able to compare those phylogenetic trees with phylogenetic trees of the diamondback terrapins that currently exist in literature in order to determine the population structure of the parasite and which host its genome more closely mirrors to determine its necessity of either species. After doing so, we'll have a better sense of these complex life cycles and how they present themselves genetically in a variety of other parasite systems, and hopefully have information necessary to conserve the diamondback terrapin throughout its range, particularly in the pockets where it's listed as endangered.